If you guys can't hear me back in the back, just let me know. I've got hit with the big giant virus, and so I've got. I'll say it right now. And so my voice may go out. I brought my elixir to help me. What we're going to talk about today is basically how cells signal. But before we do that, we need to kind of finish mechanisms of ion movement or material movement through the cell. And so the last thing we were going to talk about was osmosis, which we didn't get to. This is one of those subjects that baffles students. I mean, if you understand osmosis, I'm proud of you. At graduate school before I finally said, oh. Usually when it's taught in such a way that it's just not fun, and everyone just kind of blanks out and says, okay. Fortunately, I will ask them the test. The way to look at osmosis is just to call it diffusion of water. All right? And since we already know what diffusion is, concentration gradient, what we're looking at here is water moving down a concentration gradient, moving from an area of high water to an area of low water concentration. All right? So if you understand that, now understand osmosis. All right? We've got to make it complicated. Well, not complicated, make it confusing. What we're also doing when we're talking about the movement of water is we're usually talking about water in the context of a solution. Right? So what that means is you have solutes, little particles sitting in the fluid, and so you have to also take into account the number of particles that are in that solution. And so here we have an example, which is the standard example you've seen since the dawn of time, usually some U-shaped tube, which is divided by a semiconductor. And it's saying which way is the water going to move. Well, what I said, it's concentration gradient. If that membrane is permeable both to the solute, whatever it happens to be, and the water, what's the going to do? Move down concentration gradient, right? What's water going to do? Down concentration gradient. So and say permeable only to water. All right, and this is where it all gets ugly and confusing. Again, this moving down its concentration gradient. And so what we end up with is more water on the but what we get is equilibrium by. Right? In other words, the ratio of water to solute is the same on either side of the membrane. All right? So that's the basics. But we have a problem. We have something that throws everybody. Right? Osmotic pressure is the opposing, in other words, the hydrostatic pressure on the side that is receiving water. It's a force that prevents put it into actually understand and see. Picture a telephone booth. I know this is kind of back in the 60s, there was a really big fad that was going on. People you could stuff into a telephone booth. Somebody who did, or at least read about it in Time magazine. Now, how do you let's just say it's 8. You shove it in, all right? Another person parks in the map. In other words, the mass, the volume of people than any other person. Does that make sense? For stock, you get all your stocks in, one stock in, another stock out. All right? It's just area. That's what the osmotic pressure is like. It's the equal to the hydrostatic pressure on that side the water going in that says, you know what, we have enough water over here. You need to leave. So the water molecule goes, another water molecule comes down. What we end up with is equilibrium. The thing is we don't have a concentration equilibrium. In other words, we don't have balance. So water's still going to try to come in, but because of the mass of the water, the weight of the water, it keeps pushing the water back out. All right? So the definition, the strict definition, is the pressure of the solution that opposes water moving in. In other words, stops osmosis. Does that make sense? Is it? So I want you to actually walk out of this class with at least knowing one thing. Well, good. I like that. Now, osmolarity, the term we use to describe the number of particles in a solution, includes ions, intact molecules, both, anything that you put into a solution results in its osmolarity. All right? Now, this doesn't say anything about the actual composition of the particles. So, for example, if you take glucose and put it in a solution, glucose doesn't so every one glucose yields one particle of solute. All right? But if you put something in like sodium chloride, each particle of sodium chloride is going to dissociate into sodium and chloride. And so the osmolarity result is going to be twice the amount of what you expect for the particle. Does that make sense? So you think of this about a protein. Protein, for example, can be several thousand amino acids. Right? In the digestive, you have enzymes protein into little tiny amino acids. It starts off with an osmolarity of 1. When you start topping it up, the osmolarity begins to increase. They're just amino acids. Notice it didn't say anything about what was in solution, it just told you the number of particles in solution. Another definition that you're going to be seeing a lot is tonicity. And this refers to the ability to cause itself in or this is that picture that you saw a long time ago where the red blood cells in the solution. Now we have 3 different states: isotonic, hypertonic, and hypotonic. I know some test especially, but there's an easy way to remember this. What we're talking about when we're referring to tonicity is not really the water in the solution. We're now referring to the solute in solution. All right? So, when you look at this, tonic refers to solute. So, iso is same number of solutes. Hyper means more solutes. Hypo, less. You have more solutes, which way you want to go. More solutes, you have less solutes, the way the water's going to want to go. Away from less solutes. All right? So it's easy to get confused, but if you look at the words in terms of what they mean, you'll never be confused. More solute, that means less water. Less water means water's going to go. Hypotonic means less solute, means more water. Water's going to move away from. And isotonic is obviously the same. There's not going to be any net movement. Fun things about these recordings is all the sniffling and coughing and the hacking and the sniffling again. It's really, really fun. If you don't want to listen to me do that, go back and listen to an old lecture. It's all the same. Now, the human body is electrically. And what that means is that we have the same number of positive ions and the same number of negative ions. In other words, they're in balance. Right? Every positive charge is a negative charge. The problem is that distribution, which we saw when we looked at that slide, which shows all the different ions. High potassium on the inside, low potassium on the outside, high sodium on the outside, low sodium on the inside, and so forth. And we know, because we've learned these things, that opposites attract. Right? We've dated enough opposites to realize that. Opposites attract and there's unequal distribution. What that means is that there are things that are attracted to one another that are going to be separated. Now, if you've ever been to a high school dance, you've seen how hard it is to separate things. Right? You see those chaperones moving around, they go in between, they know you've got to dance this far apart, they say, yes sir, and the second that person turns around, they're right back together again. Right? Well, that's what's going on with body too. Because you don't have chaperones moving things around. What you have are plasma membranes. Plasma membranes. Now, technically, plasma membrane is doing work. Right? Now, when these charges move, what they're doing is what is called a conductor. And our conductors in our body, the water. The things that keep these charges separate are called insulators. The insulator is that plasma membrane. If you look at our little example here, we have in the top example everything is nice and even. But over here in the bottom, what we can see is how that distribution actually takes place. There's positive, negative charges inside the cell, but there happens to be things that are we're going to be talking about these electrical charges. Is this mismatch that finds itself surrounding the plasma membrane. So our plasma membrane is the thing that's actually doing the work. And when we have this separation, what we're calling membrane potential, we have potential energy. Formed. Alright? Where energy is used up. Now, the only place where this is occurring is localized right around the plasma membrane. So at this point right here, or if you wanted to say just right around those surfaces. Okay? Look at the charges. What we're looking at is a difference in electrical potential. What we have is what we refer to as an electrical gradient. If you have a lot of positive charges over here and less positive charges over here, which way are the positive charges going to want to go? Downstream. Alright? You can do it the same way. You have a lot of positive charges, a lot of negative charges, which way are the positive charges going to want to go? Now, it's easy to look at positive and negative, but we're also kind of referring to the movement of positive charges. Because when you're talking about current, it's the movement of positive charges. So, less positive charges means you have a lot of negative charge. Alright? The problem is that we have different types of molecules that carry both these positive and negative charges. So we have that have gradients as well. And we have to take, whenever we're talking about movement of ions, so it's not just, oh, I have a lot of positive charges over here and a lot of negative charges over here, or less positive charges. You have to take into account what are these positive charges here and what are these negative charges over here. And so you're dealing and you're dealing with electrical gradients, and the combined forces that these two things have is now, all cells in the body have something that's called a resting membrane potential. And this is the membrane potential when the cells are at rest. In other words, when cells are not producing an electrical signal. It's no current. Of a membrane potential. These are your muscles and the nervous system, and we're going to talk about what it's doing when we get to those sections. Now, the ions that are responsible for this, and it's because they're unequally distributed. We have sodium ions, magnesium, potassium, calcium, phosphate, chlorine, bicarbonate, and the PR minus is protons, or excuse me, not protons, proteins. Anionic proteins. Your textbook uses PR minus. The old textbook, the Sherwood textbook, if you have that, uses A minus. Either of those represent the exact same thing. Anionic proteins. Large, they're negatively charged, and don't have a lot of movement. Now, of all of these, and this isn't even the full list, this is just the general list. Of all of these ions, the ones that play the biggest role in membrane potentials are sodium, potassium, and those anionic cellular proteins. So what we're going to be looking at is the movement of these three ions back and forth across the membrane. Now, I'll throw this in here as well. Chlorine also plays a role. We're just going to exclude, but it does play a role. Now, what we're going to do is we're going to look at this under an, and then we're going to put it into the context of the human body. Look at potassium by itself. Artificial cell sitting in fluid that has and the anionic proteins. In the body, the proteins are going to be found in cells. Very rarely do you see cell or proteins really floating around in the fluid. You have proteins in the plasma, but because of that wall, the plasma interstitial space, proteins are going to find themselves in interstitial space. So we're talking about these fluid surrounding cells with very little to no proteins. Now, in our little example over here under A, if you look up the number of proteins and you look at the number of potassium, you'll see that they're matched. And so everything's happy and good. If you look at the sodium and chloride, everything's matched and everything's happy and good. But when you put a leak channel, potassium leak channel in place in that cell wall, now we're going to be dealing with concentration gradients, right? No potassium on the outside, lots of potassium on the inside, relatively speaking. So where's potassium going to want to go? Out the cell. Easy enough. And when it goes, it takes with it a positive charge. What we've done now is gone from a neutrally or electrically neutral condition to now where we have a negative charge on the inside of the cell. So I'm just going to do that. And a positive charge on the outside of the cell. Well, that's imbalanced. But that's okay. Now, pull the potassium back through the leak channel. And so what we have here is a balance between the concentration gradient and the electrical gradient. All right? In other words, these two forces are opposing one another to direct the direction in which these ions are going to be moving. Pull the potassium back in. And remember, it's only potassium because this is a potassium channel. You can't see it. All right? Potassium wants to move down its concentration gradient because of the natural imbalance that is existing in terms of concentration. And so we're going to see these two forces pulling back and forth against one another. And what we can do is we can actually calculate out what that potential actually, what charge does it take where we finally get balance between the concentration gradient and the electric gradient. We refer to this as the equilibrium potential. Every ion has... Yes, sir. Say that again? No. We'll go into all the little tiny details. All right. I'll point out is I do use all the movement. We're talking a couple of ions are all the time. All right. Ignore the hyperbole and just kind of understand the principle right now. So, the equilibrium potential describes that balance between the concentration gradient and the electrical gradient. Sad thing. There's a Now, the good news, do I ever test mathematical skills? No. 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 This isn't math. We know how to use calculators. As long as you know how to use a formula, you can figure out sit there and you don't have the opportunity to make it. All right? So what we have to do here is we have to understand an equation based upon what it's asking us to understand. We're trying to determine the equilibrium potential of a given ion. And the Nernst equation allows us to actually calculate this. And really all we've got to know is the relative concentration of the ion outside the cell and the relative concentration inside the cell. Plug it into the formula and we can figure it out. If we know the valence of a particular ion, that's another factor that affects how we determine our answer. So this is the value of the equilibrium. This is going to be represented in millivolts. Calculated out basically includes universal gas constant after Faraday's and then plus the log, the regular log, and then we're going to have an ion valence. Potassium has a valence of plus 1. Calcium has 2. This is what the valence. It's easy to go. All right, and then you just look at the concentration. When you're looking at it, or if you know it, and you'd be able to solve it out. So, for example, with sodium, this is just an example. Sodium has an ion concentration outside the cell of about 150, has an ion concentration of about 15 on the inside. Take the log of that. It has a valence of 1. 15 goes into 150 10 times. So a log of 10 is 1. 1 times 61 is about 61. So for sodium, the equilibrium potential is about 61. It's actually closer to 60. We'll go with that later. All right. So that's in essence how you do it. All right. It's a positive number. And so what that means is that there are more ions outside. So ions will continue to move until was 60 millivolts, or in this case, make sure I have a number that's correct before I do this. Yeah. So for potassium, all right, potassium has an ion concentration of 5 millimolar versus 150 millimolar, has a 61 log, has a positive charge, 5, that's 1/30th. Anyone want to do the log of that? I don't think so, me neither. The answer will come out to, so 61 log is approximately equal to 90 millivolts. All right. Negative 90 millivolts. So I know that is not positive. That's right, it's less. A log fraction is also negative. Right? So if you have a negative valence and you have a negative and you have a fraction over in the log, what's the absolute equilibrium of these? We don't have to do math in here because you already know how to do math. All right. But what I'm trying to do here is that we can actually calculate individual ion in a laboratory setting. We can actually figure out the equilibrium potential. And so, in our example on the previous slide, where we had the potassium would continue to move out until the equilibrium potential was minus 90 millivolts. In other words, created a negative charge of minus 90 inside the cell. At which point, potassium would start moving back into the cell. Does that make sense? Yeah.
So are you saying that's like the equilibrium that's what it's trying to reach? I'm trying to understand.
All right. No, that's a good question. So the question was, so is this equilibrium potential, is this what the cell is trying to reach? This is what this particular... So in our little example over here, I'm going to pop back. So, right. So for here, potassium will continue to move out, creating a whole bunch of negative charges, and when those negative charges on the inside equals minus 90, in other words, minus 90 as a result of all that negative charge, then potassium is going to start moving back in. All right? And it won't waver. It'll stay at minus 90 because once it gets at minus 90, the number of potassiums leaving is equal to the number of potassiums coming back in. All right? And then you could do the same thing for plus 60 millivolts. So, until the membrane potential equals plus 60. Yes?
Now, when you create potential, if there's a sodium channel, what's the top going in there and offset?
Good question. And we're going to get to it. I like it when people are thinking ahead. Say, wait a second. In laboratory setting, you're only showing ion channels. What happens when you have 2 different ion channels, or 3, or 6, or whatever? Well, then there's all sorts of things to do. But we'll get to it. I know. We don't have to do it. Someone else has to do it. All right. But do we understand the Nernst equation and how it works, more or less? Okay. And what we're trying to do is to look at sodium. So here is, we're looking at how we get to that equilibrium potential. And so I already demonstrated to you how we can actually calculate it out. And in essence, it's just showing you when you have sodium moving into the cell, what's going to happen is every time sodium moves in, you create a positive charge. And so when that positive charge is equal to 60 millivolts, then you're going to have equilibrium. Sodium is going to stop moving in. All right? It moves in at the same rate as it moves out. Now, we're talking about potentials, though. We're not talking about that one ion. We're talking about 2 ions. As I mentioned. Now, I've also talked about the sodium-potassium pump being in cells. And if we just allowed sodium and potassium to move down their gradients, eventually everything would reach some degree of equilibrium. Nothing else would happen. But we have the sodium-potassium pump to make sure that we're constantly moving the ions back against their gradient to put them back into position so they can move back down the concentration gradient. So, for every ATP, we're going to actually move and create a 2 charges. In other words, we're going to move 1 sodium back out. Well, we're really moving 3 sodiums back out, 2 potassiums back into the cell, and so we're creating a difference of charge of about +1. And so when you're looking at membrane potential, the sodium-potassium ATP pump, which is moving things back to where they started, creates about 20% of the resting membrane potential. So, now, I kind of jumped ahead. I just realized I jumped ahead here without saying something. When we're talking about moving down the concentration gradient, the reason you create this negative charge is because of the ionic protein. Negative charge protein are cells. So that negative charge wants to go, but it can't. It's stuck inside the cell, so it's slung across the plasma membrane cell. All right. Chlorine is sitting outside the cell channels, but we're not going to worry about that. So when it's leaving that chlorine behind, and all right. But again, when we look at these conditions, it was only an experiment. We only looked at when we put a leak channel for potassium or a leak channel for sodium. So under those conditions, we have those equilibrium potential -90 and +60, but in a living cell, we have channels for both. So, in order to determine what the resting membrane potential is, we have to take into account not only the equilibrium potentials of each of the ions that we're looking at, but we also have to take into consideration the degree of permeability of that ion or that membrane for those ions. In other words, we have to look at the number of channels. All right? And so for a normal cell, what we'll find is that there are more potassium leak channels, between 50 to 75 more sodium or potassium per sodium channel. Does that make sense? In other words, we have 2 sodium channels. And just by greater, right? And that's true. Potassium has a huge effect on the membrane potential. So when we're actually looking at these things, you know, look at this cell has more permeability for this ion. There's the equilibrium potential is going to be and more like this particular ion. So in looking at a nerve cell, if we know -9. Right. And in fact, this is actually true. The membrane potential of a neuron is -70 volts. Now, I'm not going to ever ask the question, what is the resting membrane potential? What is the resting membrane potential of a cardiac? Numbers that you memorize. But it's kind of a good idea to know the -70 just because it's going to show up over and over and over again. I'm not going to ask you the question, but it's kind of nice to have as a baseline. All right. Now here's the -70 millivolts as a resting membrane potential. Does it satisfy the equilibrium potential? Does it satisfy the resting membrane? Satisfy the equilibrium potential? So what's sodium going to do? It's going to keep moving into the cell. Will it ever satisfy the condition? No. Why? Because we have sodium, potassium, ATPase. So what we have here in our cells is a constant movement of ions down their electrochemical rate, never being able to reach the equilibrium potential for either ion, and so we're having this constant movement. All right. Neurons sit at -70 at all times. Seeing a lot of faces doing this. Where's he going with this? Now the -70 is going to favor potassium moving out. The -70 favors sodium moving in. All right. That's in essence what we're saying here. And because, even though we have more leak channels, because we have that ATPase pump, pumping things back and forth, will never satisfy both conditions, so things are constantly in motion. Now, back to some definitions. I think it's the way that your book does this and the way that I do this, because I'm following your book, kind of interrupts our train of thought. I want you to just kind of put equilibrium potentials on the back burner for a moment, because we're going to go and move to signal transduction, and then on Thursday we come back to all of this stuff. All right? But before we do that, I want to just kind of mention the different types of electrical systems. Yes, sir? In this particular case, this is inside the cell, and this is outside. No problem. That's good. It's not always clear on the slides, and the figure I use. Flip back and forth. All right. So this is just definition stuff. Now I need you to think back to 3rd grade. I think it was 3rd grade. I don't have really good memory at this point. You remember when you used the line, you had to figure out how far it was. It said you're 10 to the left. So what we're really dealing here is we're basically learning how to deal with negative numbers and positive numbers. I think that was 3rd grade. All right. What I want you to do when you're looking at these things, what we're talking about here is a... All right. A resting membrane always going to be -70. All right. It's going to be some value other than 0. Whenever it's some value other than 0, we refer to the membrane as being... Does it make positive? -7, polarized. 0 is not polarized. All right. So any state of the membrane where the membrane is a state of polarization. So using our neuron here at -70, we are in a polarized state. When you move towards 0, depending on where you are depolarizing. You're moving away from the polarized state that you were. You're less polarized than you were when you started. So in this case, going from -70 towards 0 is becoming polarized. But if you're up here at +40, and you started going towards 0, that is also depolarization. So you always have to remember, where am I in the context of my resting? All right? Now, often we're dealing with looking at remembering depolarized means less polarized than before. When you return back to a resting membrane potential, that's referred to as repolarization. If you move further away from 0, that's hyperpolarization. So that right here, moving back up, would be repolarization, because you're going back to rest. All right? So hyperpolarized, further away from 0. Going back to hyperpolarized. That's how we use these verbs. Ways they can do it either electrically or they can do it chemically. When we're talking about electrical communication, what we're doing is we're changing the membrane potential of a given cell. When we're talking about chemicals, we're talking about a molecule that's secreted into fluid and are influencing how a particular cell behaves. Using either of these two methods, we can communicate either through gap junctions, through contact, through local signaling mechanism, or we can do it in a long-distance fashion. And we're going to look at each of these 4 types. The gap junction is basically what we have are little tiny gaps between the cells. Hence the term the gap junction. Gap junction is formed by a particular protein called a connexin. There are many different types of connexins in the body. I think there's roughly 20 of them, probably more now that I think about it. And what we can do is when these two cells are connected to one another by these connexins, we've created this little passageway so really small molecules can actually travel back and forth between them. This includes ATP, amino acids, chemicals, ions, any of these things, little tiny chemical messages, as long as they're small enough. Any move back and forth between the connected cells, thus resulting in a change in one cell relative to the other. This is how cardiac muscle works. They're connected by gap junctions, and they through the cardiac. The other cells, chemicals as well. Contact-dependent signaling, on the other hand, is a cell recognition mechanism. All right, you'll find on one cell is a receptor. When those things come together, they're communicating. This one's telling the receptor, this cell, you need to behave in this fashion. Molecules are referred to as cell adhesion molecules, the CAMs. All right? Yes.
Is that why cardiac
That's exactly why they do. Because the ions move very, very quickly through. All right? So, in the contact-dependent signaling, we're basically having a direct contact, and we're using cell adhesion molecules, or the CAMs, in order for communication to take place. Autocrine signaling is when a cell releases some molecule, and that molecule, instead of traveling to some distance, comes back on and binds the receptor. Seems kind of silly, doesn't it? I need to tell myself what to do. Yes, sir. Uh-huh. Now, probably be a good idea. If you wanted to beat me to the punch and look up neutrophil and diapedesis. That would be one of the two things, and it actually shows you sneak through the capillary walls. How do you spell diapedesis? D-I-A-P-E-D-I-D-E-S-I-S. Diapedesis, yes. I will never win at a spell. Yeah, and yeah. All right. Now, why would you want to do this? Well, it's one way that you can actually regulate interaction inside or a pathway. And in fact, it actually occurs in the reproductive tract after ovulation. I know you guys, we haven't talked about this. You basically go from, in ovulation, you go from FSH-mediated activity to LH-mediated activity. And so a lot of the same molecules are involved, but to make that switch, you're basically going through this sort of pathway where the cell itself says it's time to change activity. So it tells itself what to do. Paracrine is when a cell releases a factor and it affects nearby cells. All right. So basically the cell is saying, this is what I'm detecting, and this is how you all should behave. Huh? Paracrine. Interference would be a good example of that. Absolutely. Now, in either of these two cases, what's the molecule doing? Basically, it's moving through the interstitial fluid and traveling a very, very short distance. It's not traveling far throughout the body. It's limited to its movement. In terms of long distance signaling, at this point, what we're going to do is we're going to take that chemical message and release into the bloodstream. And at this point, it can travel throughout the whole body. But it's not going to affect every cell throughout the whole body. It's going to affect the receptor. So in our little example here, we have a cell with the receptor, a cell with the receptor. The cell without the receptor does not respond to the signal. The cell with the receptor will respond to the signal. You'll get a response. All right? Both the nervous system and the endocrine system use long-distance signaling. Chemical message is often referred to as a hormone. You might also even use many of these different types of factors. All right? But hormone is like the classic example of this type of signaling, a chemical message that travels in the bloodstream. Now, the nervous system also uses electrical signaling. All right? And what we're doing here is we're changing the membrane potential along the length of a neuron, and that electrical signal will then be converted to a chemical signal. The chemical will then be released at the terminal end of the neuron, travel a very short distance across a space called a synapse, and it will bind to a receptor on the target cell, thus creating a change in that. This chemical signal is referred to as a neurocrine. And actually, that's the catch-all phrase. And it could be a neurotransmitter, a neuromodulator, or a neurohormone. Now, generally speaking, I'm going to start with the neurohormone. A neurohormone does not act directly on a cell across a synapse. Neurohormone basically goes into the bloodstream, much like a hormone. The difference between a hormone and a hormone is the origin. Nervous system. A hormone of system. All in the structure. The neurotransmitter is that chemical signal that moves across to create a rapid response. All right. Generally speaking, it's causing a change in the membrane potential by opening up a channel. The neuromodulator, on the other hand, is similar to paracrine signaling that we saw a little bit earlier. Here, we're going to be dealing with a protein that is going to act in a paracrine fashion to change the behavior of that particular cell over a longer period of time. It's a sustained response, and it's going to be a modulated response. In other words, where we're getting a very rapid response here, slower response with the neuromodulator. Does that make sense? Or neuromodulator? Modulate the response. We're going to change the characteristic of the response. So in other words, in a brief example, let's a very rapid response. What a neuromodulator will do will change the nature of the cell here. So it takes longer for you to get a response out of it, but it's going to change the nature of, say, the neurotransmitter. As an example, that's just kind of stupid. When it's cold outside, you go outside and you start shivering, right? That is your normal response, right? If you put on a coat and go outside, will you shiver? No, that's modulating your response. So a neuromodulator, stupid example, but it kind of gives you the idea of what modulation means. Yes, sir.
Changing the cardiac cell or the...
It can either act one way or the other. It can act in an autocrine fashion and tell the the neuron that's releasing the neuromodulator to change its behavior in an autocrine fashion. The heart cell and change how it responds. Now we're not telling you how. We're not telling you how it changes the response. We can put in more receptors. We can take away receptors. Yeah, we can well, we won't block a receptor in this sense, although that would probably be another neurotransmitter. All right, but in essence, we're taking what is a normal response. Well, we're going to see examples of this when we get to the nervous system. That's correct. They're not permanent. Right. It's changing something for a short. For a period of time. All right. Whereas, for example, if a neurotransmitter were to bind to its receptor, you're going to get a very, very quick response, right? But let's say you don't want quick responses. Let's say you want slower responses. Well, you release a neuromodulator to change, for a short period of time how that cell responds. And so instead of getting a quicker response, you may get a slower response. But you don't want that to be permanent. You want it to be able to adjust based upon your need. Kind of a way to regulate how the cells change those relationships. Now the rule for pathways is pretty simple. 5-step process. The thing that a signaling pathway is going to be dependent upon is the presence of the receptor molecule. If you don't have the receptor, you can't respond to the chemical. All signaling pathways has 4 features. First, you're going to have the signaling molecule. We refer to this as the ligand. It is carrying the message. It doesn't mean that it has something that's holding up the arm and creates the cell. All right? The chemical itself is the message. All right? So it's going to have a specific response in that cell based upon whatever that signaling molecule actually is. But what dictates that relationship is the actual receptor. When the signaling molecule comes along, it binds to its receptor. This activates the receptor and initiates an intracellular pathway. Very often, the example that we're going to be looking at one pathway because it's simple. All right? But don't think that that's how it's working. Really, you can think of it activating 2 or more pathways at the same time, making it complex and hard. It's simple. All right? So, signaling molecule binds to the receptor. Receptor becomes activated, activates the molecule. Inside the cell, that signaling molecule then activates this pathway, which results in either protein synthesis, all right, referred to new protein or de novo protein synthesis, or it modifies proteins that already exist inside the cell. In other words, activates or deactivates cell or proteins that are already there. So it changes the actual function of the cell by either creating new proteins or changing what's turned on and what's turned off. If you walk into a room and someone says, hey, turn off the light, and you turn off the light, Psychological? Yeah, start. That's what you've done. Someone told you that there's a signal, you are the you turned it off. That's what we're talking about here. Cell's still a cell, room's still a room, but it's now dark. Now there are two different types of chemical signals. We refer to them as being hydrophilic or lipophilic. Or if you want to, lipophobic and hydrophilic. But we'll just keep with the first, hydrophilic or lipophilic. Definitions are pretty straightforward. Hydrophilic molecules are water soluble and or poorly lipid soluble. They're primarily proteins or peptides. They bind to receptors found on the surface of the cell, so membrane-bound receptors. That would make sense. They're water-loving. They can't penetrate through the membrane, so they have to bind to something that's on the surface. Generally speaking, we change the activity of proteins already found in the cell. Some examples, which you may or may not know, binds to the surface receptor. Catecholamine, adrenaline, for example, FSA, follicle stimulating hormone, all surface already in existence in the cell. Lipophilic, on the other hand, will penetrate through the plasma membrane. They do not like water. All right, so they easily diffuse through the plasma membrane, and they bind inside the cell. That's what this is right here. Supposed to be, at least. Now, generally speaking, these types of receptors are transcription receptors, meaning they bind to DNA. And so when these types of receptors by their ligand, lipophilic molecules, they bind to DNA and or the transcription of new, which results in new proteins. So we make new proteins in general with lipophilic molecules. Intracellular, new proteins. Examples include the steroids and thyroid hormone. So far, everyone with me? Okay. Now, there are many different types of membrane proteins. We have ligand-gated channels. For example, the ligand binds to the channel, causes the channel to open so that ions or whatever can pass through. Another example would be the receptor enzyme, where the receptor gets bound by the ligand, activates an enzyme, so you start changing the activity of molecules inside the cell. We're going to look a little in depth at the G-protein coupled receptor. Here, we bind to this G-protein coupled receptor, which is coupled to a, and the G-protein becomes activated and changes the activity of the enzyme down. The last is the integrin. As an example, the integrin, when it's bound, causes a change in the cytoskeleton. One of the labs that was next door to my lab, when I was a graduate, worked on the integrin. One of the they did stuff for NASA, and they wanted to look at zero-G, what effect zero-G had on the integrins. Really kind of cool. Cell plates with these cells, and they treat them with different types of integrins. But many of these cells were cells that were mobile to begin with. And so you get to watch to see how they would try to move. And it's kind of interesting. It was like watching, they basically kind of crawl over themselves like so, and moving the integrins themselves. But that's an example of cytoskeletal. The process by which we're going to be activating this downstream cascade is referred to as signal transduction. Now, signal transduction really means taking a signal molecule and transducing another thing. All right? In other words, what we're not doing is we're not putting it inside the chemical doing something. The chemical is activating something, which activates something else, so on and so forth down the line. That's the transduction, right? Taking one, converting it into a different signal. That's what transduction means. We often refer to signal transduction as second messenger systems. The first message in a second messenger system is the initial chemical messenger. In other words, it is the hydrophilic molecule that has been released and is binding to the receptor. The second message is a molecule downstream that activates what is called the effector. All right? It's not necessarily the next molecule in line, be down a little bit further, and we're going to see that in just a moment. All right? It is the one detecting. So what we're doing is we're taking a chemical signal and we're converting it into a response. And very often, every time a chemical is bound to the receptor, what you're going to see is an amplification. In other words, when we activate the receptor, it's going to be a ratio. One single response molecules. And then those 10 molecules might cause a change in 10 other molecules. And those 10 molecules go down the line. We refer to this as the cascade effect. The benefit to this is how much hormone we can actually release to cause a massive effect. All right? The other benefit to this is that because you see an amplification process along the way, you can actually multiply your response through several different pathways. Now, like I said, we're going to focus on a single pathway, but instead of one molecule just activating another molecule downstream, you can think of it as this molecule activates 3 molecules, and this molecule here activates 3 different molecules. Activated a whole bunch of different systems simultaneously. Does that make sense? Okay. And so this is kind of what it looks like. You take the initial stimulus, you activate one molecule, which activates another molecule, which activates another molecule, and the end result being the product that you're trying to do. Now, the receptor enzymes, what they do is that you have a receptor on the extracellular fluid side, you have an enzyme region on the intracellular fluid side, or intracellular fluid, and what you can do is when you stimulate, you activate that enzyme, and so you can start producing new products directly. And so an example of this would be the protein tyrosine kinases, which take the protein and phosphorylate them. So what we've done is we've changed the nature of the protein, depending on the protein that's down. So this is either a new active protein or a new deactivated protein. Sometimes a receptor enzyme complex can be different proteins in and of itself. In the example, like a little cyclase, and if you don't know what that is, don't worry about it. I'm not going to ask for examples. But I'm interested in the G proteins, and I'll kind of go through all of these in just a moment. But the G protein, what we have is the G protein is a transducer. All right, it's the one that's actually converting the message. And the G protein has 3 subunits to it: an alpha, beta, and a gamma subunit. The beta and the gamma and the alpha are all bound together in its inactive form. But when you bind the G protein coupled receptor with the ligand, that activates the G protein. And then it goes and activates some enzyme down. In our example, what we're going to be looking at, all right, so and then after it binds to, cleaves the GTP, it's in the energy phosphate group, becomes inactive. And when it's inactive, it's no longer happy, so it goes back and joins up with the gamma stuff. So it turns itself off after it activates the cyclase. So bind, activate, activation, activates something new. Now this is the first step in the classic cyclic AMP messenger system. Signaling molecule binds to the G protein coupled receptor. G protein coupled receptor activates the G protein. The alpha subunit traverses away from the beta and the gamma subunits, activates adenyl cyclase, Adenyl cyclase takes ATP and converts it into cyclic AMP. Cyclic AMP is our second messenger. Cyclic AMP binds to a protein called protein kinase A. Protein kinase A is a molecule that can change the activity of another protein. All right? It will add a phosphate, activate, or in some cases deactivate the protein. So, the phosphorylation event changes the activity of the proteins at that level. When you change the protein activity of those proteins, you're going to change another level of proteins, and another level of proteins, and another level of proteins, and so forth. And what you've done is you can create it. Does that make sense? That's a good question. Why? Because each cell has a job, right? Salivary gland for a second. Cell in the salivary. All right. You want to be salivating all the time? No. Well, actually you do, but do you want to be salivating a lot? No. All right. Is there times when you want to? When? When you want to eat. When do you want to salivate with? When you're talking. Yeah, good. That's all right. And so, as an example, you have. I'm just using this. I'm not saying second messenger system, but you basically have a state of the cells: produce saliva, produce less saliva. All right. One of those two states. All right. So, using a second messenger system, what you can do is you can change those two states. Now, I'm not saying a salivary gland necessarily does that. All right. It's just an easy example to show you two different states. And that's in essence what you're doing. You're changing what the function of the cell is by using the chemical to change what's already there. Turning things on and turning things off. Changing the activity. That's a good question. I've never had anyone ask me that before. Oh, no, that's, huh?
I have another quick question.
Yeah, go ahead.
Where does the G protein get the ATP from? Is it bound sitting there as potential, or is it?
All right. So where does that? Well, so remember, in every cell you have a certain amount of ATP. All right. So in terms of the G protein, remember deals with GTP and GDP. Again, you're going to have ATP being produced. It's always available, or I shouldn't say always, but it's readily available. All right. ATP and the GDP, we're just kind of ignoring as to where they come from. We're just saying that we know that the cells have this. All right. As far as ATP, the same thing. We know that we're producing ATP, just a function of normal glucose metabolism. ATP is there. Take that ATP, we're going to change its molecular structure, get rid of 2-phosphate groups. Cyclical molecule. Cyclical molecule binds to protein kinase A, and because protein kinase A actually activates protein kinase A. All right. So that's why cyclic is a messenger. It's what activates the effector. So in our little system here, protein kinase A is our effector. This is our second messenger. And it should kind of give you a hint. Cyclic AMP, second messenger system. Okay. Now this is classic. Say 80% of the signaling transduction pathways in the body use some sort of modification of the second messenger system. It's the first one discovered, and it's obvious why, because there's so much of it. There's another type that's actually classic as well, and this makes up the next greater portion, and this is calcium calmodulin second messenger system. All right. Now, again, we have a ligand binding to a G protein coupled receptor that activates a G protein, but G protein, instead of activating adenyl cyclase, activates another molecule called phospholipase. Phospholipase C takes a membrane phospholipid, PIP2, and cleaves it. And it produces two molecules, IP3, triiodo, well, I'm not going to be able to remember, IP3 and diacylglyceride. All right? Thank you. Diacylglyceride is a second messenger, and it activates an effector called protein kinase C. Protein kinase C then phosphorylates a whole bunch of proteins like protein kinase A did. Right? You get a change in the activity of the cell. IP3, on the other hand, binds to a receptor on the plasmic reticulum, resulting in the opening of a channel. Calcium is released out into the cell. Calcium binds to a molecule called calmodulin. Calmodulin is like protein kinase A, and it's like a bunch of changes in the cellular response, or in the cellular activity. So we have in this system, we have two different messengers. Right. We have DAG, and we have calcium. Now, why is an IP3 a second messenger? If you had to take a guess. Because what? Because it's binds to a receptor. It's not actually acting as an effector, but it's very similar to that. What it's doing is opening to allow for the chemical something. All right? So that's why we don't say IP3 is the second messenger here. Right, it just opens the channel. All right? Now, you're looking at this, what you're thinking. You're going, oh my God, so hard. What I want you to do is I want you to go back and take a look at the calcium channel, or the calcium, excuse me, look at calcium calmodulin, and compare it to cyclic AMP. Is this the same? Yeah. Is this the same? I should actually even circle this. Is this the same? Yeah. So basically, upstream has the same mechanism. Downstream has basically the same mechanism. It's the stuff in between different. It happens to be a different mechanism that the cells have developed to allow for second messenger systems to work. Right? So not everything's under control of cyclic AMP. And it's not real hard. You have one, this one goes here, that one goes there. There you go, 2 steps. Now, soup gets confusing, especially since everything is PKA, PKC, PKLC, and just wait if you go into biology or molecular biology, you'll have PK. Phospholipase. There's a D and an E. You know, so I think there's alphabet soup. Principle down, you'll understand, oh, phospholipase, what does it do? Takes a phospholipid, and it's an enzyme because there's ACE on the end, and it cleaves the phospholipid. Where do we find phospholipids? In membranes. So it's really not that tough.
Neither DAG and IP3 are.
DAG is, diacylglyceride is, because it activates PKC. And PKC is an effector. So here's our effector. And down here, it's not showing you, but you put calmodulin down here. And calmodulin is our effector. Now they're activating 2 separate pathways. So this is an example of 2 pathways using a single message. And why it's complex and scary. So calcium increase. I'm glad you pointed that out because that's oftentimes. Calcium is being stored in the endoplasmic reticulum. In other words, in the cytosol itself, we have very low levels. Open up the endoplasmic reticulum. Here's my hyperbole. Rushes out, and so we've gone from a very low concentration to a much, much higher concentration.
Is it the concentration that activates the calmodulin, or is
It's the calcium itself. Calcium binds to calmodulin and then causes the change in the shape of the molecule, which then activates it. Good question. Last one is the receptor channel, which is very, very easy, very rapid response. The diagram is trying to show you that there's actually multiple ways to do it. All right. When you activate a receptor channel, you're basically taking a ligand and you're binding to the channel itself. Permeability of the membrane. All right. So ions can actually come rushing through. Active G-protein coupled receptor, for example, and activate it through an intracellular mechanism. This is just another downstream target for the second mechanism, or could be. We'll see this when we look at cardiac tissue. This is one of the ways that cardiac affects their ion channels. This is the scary map that you shouldn't go, oh, I've got to memorize all this stuff. No, this just actually throws all of them. All right? What do they do? In a one way or the other. We see a whole bunch of different activity at the bottom. Good news, this is the last slide. Yay! All right. I don't want to reiterate it so many times that you get bored of it, but I'm going to say it one more time. The response in the target cell is determined by the receptor. It's not determined by the ligand. All right? In other words, what receptor is found on the cell is going to determine what is going to actually be activated. All right? Down here we have an example of epinephrine. Epinephrine has two different types of receptors that it can bind to, an alpha receptor and a beta receptor. When it acts on an alpha receptor, the response is dependent upon, not epinephrine, it's dependent upon the receptor, the alpha receptor. It results in the vasoconstriction of a vessel. If a beta receptor is there, the response is different. It's actually opposite. You get vasodilation. So it's a signal that tells the signal is there to say, time to do something different. Now, very often, there are ligands that look a lot like each other, and they can bind to the exact same receptor. So, every receptor is going to be subject to the rules of specificity and the rules of competition. We have terms for these types of molecules. We call something an agonist, that is a molecule that can actually bind to and turn on the receptor, or activate the receptor. An antagonist is the molecule that blocks the receptor and prevents its activity. The other thing, just to make things more complex, is there's no such thing as a single receptor. If I say FSH receptor, you don't just say, oh, okay, that's good. There's one FSH receptor. There are isoforms to many of these receptors. And depending upon the isoform, you can bind to different types of ligands, and you can get different responses based upon which isoform is actually there. And the last, you can actually modulate the responses. Like I said, with the neuromodulators, modulation can actually take place. And we have different terms that we use. One is called downregulation, one is called upregulation, and these two things should be pretty simple and straightforward to understand. You downregulate, the modulator comes in and causes a loss of a, well, excuse me, that's more desensitization, but basically what's happening is when you downregulate, you're losing the number of receptors that are available. And generally speaking, it's a result of an abnormal concentration of ligand. What that means is that the ligand itself serves as itself is served as the modulator. And that's, if you have a lot of a particular type of hormone, causes a huge response. You don't want lots and lots of huge responses. That's a bad thing. So what happens is recognize that what bonding a whole bunch of down the receptor I have. So it's unregulating on. Upregulation would be just the opposite, where the modulator comes in and causes an upregulation of receptor. In other words, abnormally low concentration of ligands. All right. Another type is desensitization, and what we're dealing with here is when the chemical actually causes a diminishing response. In other words, what's going on is downstream, even though you have the same number of receptors and the same number of ligands, the response downstream becomes diminished because you're actually regulating something downstream. all right, well, we're not going to deal with this stuff, but I'm not going to let my cell respond in the same fashion. If you don't understand desensitization, just using a poor example again, not of molecules, but if I keep hitting you on the face, hitting you on the face, and hitting you on the face, and hitting you on the face, eventually you're not going to notice me hitting you on the face. All right? You become desensitized. People walking around just, yep. So these are some examples of the ways that we model. Oh, I forgot. There's one more slide, and this is actually pretty simple. Now, it's how to turn it all off. All right? We saw that we have multiple steps. Response. In order to stop a second messenger system or a second messenger cascade, you have to stop every step that you started. So the first step is to remove the first messenger. It's really easily done because you have enzymes that are there, or you have something that can selectively uptake that molecule and destroy it. So the first messenger is removed, and so you basically terminated that first step, and so you're not going to be continually activating the system. Because this is a cascade, but because we see an amplification, we have to keep killing every step. Now the good news is at the second level, when we're dealing with the G protein, for example, we have an alpha subunit that actually turns itself off. Keep alternating between active state, which is GPP, and then we inactivate ourselves when we cleave it to inorganic phosphate. So the second step, self off. Down further, that's a little bit more complex. And so each of the downstream effectors, each of the molecules that become phosphorylated, dephosphorylated, all the things that have changed, there are molecules that are already in place that are looking for those molecules in their opposite state. So for every And so what we're really basically doing is we're putting everything back into its original state. And while this takes a little bit of time, you can see why. We keep turning on a whole bunch of stuff, then we have to turn a whole bunch of things off. But eventually you're going to reset the pathway over, reset the whole signaling pathway so that when the next molecule comes along, you can activate every single solitary step. So far, so good? All right. Now, I know everyone's running out of here, but I don't know if you heard. Today is the first day to actually register for the exams, starting at last night at midnight. You have 2 weeks till the first exam.
We got the perfect time and everything.
Perfect.
I have a question.
Uh-huh.
So,
Let me